Hey, my name is John McFetridge, and welcome to my talk on the AI meets drug design. And thank you for inviting me in to share, so I can share with you this exciting field. I've been a long time software developer, but I've always been also interested in genetics and in particular and biology. And here in my old age, I have an opportunity to combine both. And now I'm not a PhD, but I've spent a lot of time, and I must say it's just time that I've so enjoyed because this field is incredible. What's happening here? So I've been mostly using tools like Gemini and Notebook LM and YouTube, and where I can dive into as deep as I want to. And I don't have a PhD, but I think I want to share with you what I've been discovering, and I certainly have been enjoying it, and I think you will. I hope you will too. So, what are we going to discuss? Is how do we use AI to redefine drug design? We all know a little bit about AI changing the world, but this may be the biggest impact that AI has. Um, we've got to look at the drug design problem. We discuss what are proteins and what the relationship between them is in drug design. We're going to look at a couple things like Alphafold 2 and 3. And finally, um, interesting and maybe scary subject, protein design. So what's the problem? Drug discovery productivity has declined exponentially over the years. We've gone from producing in 1950 way more drugs than we do in 2020. And at the same time, and the costs have gone way up. So now it can take up to 10 years to produce a new drug, and it can cost nearly $2 billion. Um, but at the same time, the interesting thing is that our CPU and GPU power has been greatly increasing. There's no comparison. So what drug design process? Well, historically, it was all trial and error. And instead of knowing exactly which protein or gene they were targeting, in fact, they had no idea. Even Mendel didn't know what a gene was. So scientists would apply a compound. And through serendipity, many famous drugs like penicillin were discovered. And then we have natural products where traditional medicine often served as a starting point. For example, aspirin was derived from willow bark. So we often went to guys like Medicine Men, Merlin the Magician, looking for solution. Then in the 60s, we entered in this period of rational drug design, which is target based. And since the 60s, the process has shifted toward the reverse approach. Instead of looking for a drug first, the scientists identify a specific biological target, usually a protein that is responsible for a disease. Now, in the drug design process, we have various stages. The first one involves biology, my favorite. Then the second one, some chemistry, and finally execution. And in one, we have a target discovery. We're looking for the gene or the protein that's causing the issue, and we're going to validate our target, and then lead up to make optimization, where we're going to the goal is to make the drug better and better so that we can actually put it into the body and ensuring that the drug is going to hit only its target. And this can be the most expensive and time-consuming phrase. But what we're most, and then we go on to our preclinical, which is animal-based, and then our clinical trials with humans. But we're only going to look at this part here, the 1 and 2, and try. And the idea here is to understanding how proteins and molecules implicated in disease interact within the cell. It's going to give us clues as to how they function and how to modulate them. So the key is how to modulate these proteins. And this allows our chemists to test designs in silico as much as possible. So we want to drive this step in silico as much as possible. And we have to um, just a couple points here. 90% of our candidates fail. And now, what are these protein things? See, the proteins are associated are going to be associated with our drug designs because they are the target for our most of our drugs. The proteins, we've got to understand, are the building blocks of the body. They're molecular factors that support everything in your body, and they're the primary functional units of every cell. They're and they make up to 16% of your body. They're responsible for everything from building muscles to digesting food. Now, when a protein breaks or misbehaves, disease results. So our drugs are going to be targeting these proteins. We have to understand that the proteins before we have a chance of developing a new drug. So in the humans, we have 37 trillion human cells, each with 30 million protein molecules. So how are these proteins built? Well, in the body we have chromosomes in the nucleus of each cell, and in the nucleus, and these are just a string of nucleotides. Nucleotides are adenine, thymine, guanine, and cytosine. And they're going to make up, and on the chromosome, they're going to have a genes. And genes is just a series of nucleotides that are going to code for one protein. So, but the chromosome is too big to leave the nucleus. So it gets transcribed by RNA polymerase into a messenger RNA. And this is only one, and this is double-sided, this is only one-sided, and it's small enough to leave the nucleus, and it doesn't last a long time, which makes it ideal for making vaccines. And then it goes to the ribosome, and in the ribosome, we're got each of these three nucleotides will code for one amino acids. And in our body, we have 20 different amino acids. So we build up these chains of amino acids, but it's not these series of amino acids. It's they immediately fold into a 3D shape, and that 3D shape is what determines the function of the protein. So, again, for drug research, we have to understand this 3D shape. And here's just a few of what these different proteins might look like. These are really small. These things measure 3 to 6 nanometers. But the key thing is, and we got to wrap our minds that 3D shape determines the function. So for drug design, we must understand protein 3D shape. And but it's not an easy problem. In fact, this is one of the real challenges in biology in the last 50 years. Levinthal's paradox said a protein folded by random trial would take longer than the age of the universe to find its shape. So, and so doesn't some of the structure informs function. So if we don't know what these proteins look like, it's really hard for us to work out how they work and to work out how to modulate them. And almost all the drugs that we have target proteins. So a drug simply binds to a protein to break it or to stop it from doing what it normally does. And in many cases, it can be a cancer, which we need to turn it off because it doesn't have any pedal to stop it. So over the years, the last 50 years, this is as I said, one of the challenges of biology. And we've developed these machines, the X-ray crystallology, and here the the scientists first coax millions of proteins to line up perfectly into a solid crystal. And this is often the hardest part. Then they blast the crystal with high energy X-rays. the atoms in the protein scatter and diffract, and creating a distinctive pattern of spots on the detector. And then scientists work backwards and create a mesh of the 3D atomic structure. Now, this is very accurate, and the machines sort of look like this. Or we have cryo electron microscopes. They have the same problem that you have to make high quantities of ultra pure protein. And we're going to freeze the protein this time, and we're going to image it using electrons, not X-rays. This was involved in the Nobel Prize in Chemistry in 2017. But these have two big problems. They take a long time. It can almost take a year or two to come up with the 3D structure of one protein, and they're expensive. So, but over the years, we were able to come up with a 140,000 entry, 140,000 proteins were mapped by 2018, and of the 200 million in existence. So we had a ways to go. But whenever these were mapped by those one of the two previous processes, they were put it into this protein data bank, and these are the exact XYZ coordinates of the protein. But now, of the 20,000 human proteins, only 1,600 were completely mapped. So, again, what do we have to understand for drug design? Have to understand the structure of proteins, because again, function, shape equals function. But we weren't doing so well. So there were efforts to find a computational methods began, because in silico, where we do this work on with software and hardware, and we can do this. We want to do as much as possible in here to do almost this the digital twin of biology. So we're going to have like with architecture, we want to have a digital twin in the software and do as much as possible here. But the real step change and the real progress in human health is you basically want to do research and hypothesis search in silico. That's hundreds and maybe thousands of times more efficient than doing it in a wet lab. You save the wet lab just for the validation state. But as you can see, we weren't doing so good. And when you look at the results here, this is actually John Molt came up with this competition in 1994 to try and find a computational way to solve this problem. And twice every 2 years, the competition scientists would get together and they would try and solve a novel problem. But it was a problem the world wasn't very good at. So by 1994, you can see we were under 40 here. Now, John assigned, to be successful, John assigned we had to get 90. So you can see that over the years, we weren't very good at this. Well, this is back in the darker ages of AI. AI was kind of being laughed at, and nobody was able to come up with a good solution. In fact, by 2008, it started to decline. However, by 2018, things were beginning to change. AI was getting better and better. And out came DeepMind. From DeepMind, it came up with Alphafold. Now, Alphafold think of Alphafold as a large language model for proteins. And DeepMind was created by Demis Hassabis. He's still the CEO. These guys went into the competition in 2018 and they won it. And they won it, but they only got a score of about 70. And at this time, so they knew they'd won the competition, but they hadn't achieved the goal of getting 90. So they went back to the drawing board and Barney and John Jumper, and then the team in 2020 they came up with AlphaFold 2. AlphaFold 2 changed everything. it won the competition, and it got over 90. So John Molt declared the protein folding problem solved. Now, um, again, so what this does, it takes in a string of amino acids, and then it has two databases. One, a genetic database. Now, what and then the protein structure database, which is the those 200 those 170,000 that have been structures that have been laid out through the X-ray methods. Now, it's going to search this database first because it can find one that's similar. It's going to save itself a lot of time. This is interesting what this genetic database does. It's called the MSA. This is kind of like this is the blueprints of the protein, and this is how these blueprints mutate over time. And well, what's mutation got to do with anything? Well, it's here. Well, let's say we have this chain of amino acids. Pardon my wonderful drawing here, and we have one, two, three, four, five, six, seven, eight, nine of them, and two, and 10 are mutated. They're found to mutate together. Well, if they mutate together, we know they must be close to each other in their 3D structure. And the reason is that if they don't mutate together, then the protein's going to lose structure. And losing structure, it's going to lose function. Structure equals function. If it doesn't continue functioning the way it was, then it's going to probably cease to exist. So there has to be a little loop between these two to bring them together. And then it's passed to the structure module. The structure module takes the refined models and performs rotations and translations on each of the amino acids, revealing an initial guess of the 3D protein shape. And it then pumps out this 3D diagram look. So and they predicted 3D protein structure. And these results are highly accurate, often rivaling the experimental methods. But it also assigns a confidence score. So here, the green is high level, and the light blue is not so confident. So this changed everything because now the Alphafold database expanded the world's knowledge of structures 1,000-fold. So in 1 year to 214 million. Almost the scope is almost everything. It covers nearly every known protein from every organism on the earth that has its genome sequenced. And with the human proteome, it covers roughly 98.5% of all human proteins. Now, what good is this? Well, we now have mapped out 200 million, and of course, 98.5% of all the proteins in our bodies. And this again is important because structure equals function, and we now have at least approximations of the structure of all the proteins in the body. And now, previously, a PhD student might take 2 to 3 years solving one structure, or a research scientist in a lab. But now they can download a structure in seconds or have one created. And I will show you soon how they do that. And then they can spend the next time. Now, the other problem it solves, one of the issues with drug design is discovering what's called a pocket hunt. And we're going to look for these gaps or these surfaces where we can bind our small molecule drugs to. And with Alphafold, we now have there's so much, our universe of known protein structures is almost complete, greatly enabling our pocket hunt. So just as one case study, where we can accelerate the hit identification, one of the most cited successes is the work of in silico medicine, which used AlphaFold 2 to target a liver cancer. I won't try and pronounce that. Uh, now, the problem, scientists needed a hit molecule for a novel protein target, CDK20, that had no previous known 3D structure. Big surprise, because we only knew about 170,000, and in the human body, we only needed we knew about so few. But now we could, with the researchers, use the AI predicted structure to design a potential drug, because we had the tool to generate that. And the result, they identified a potent hit molecule in just 30 days. But there's a big but. This is only for proteins, and this only gave us a shape of proteins. Sure, it did a lot. Now we can understand the so many more protein, the structure of so many proteins in the body. But now, um, but it only dealt with proteins. Proteins do not exist in isolation. They exist. They are they exist in biological systems. They and they're part of bigger molecular machines where the proteins are going to interact with RNA, small molecules, and of course multiple chain proteins. So we need a way to be able to show the interaction of all these parts. Thus, they came up with DeepMind came up with AlphaFold 3. And so here, the goal is to reduce the need for physical testing of candidate drug compounds by enabling high fidelity molecular simulations that can be run entirely in silico without incurring the prohibitive costs of traditional chemistry methods. Now, this is just briefly what AlphaFold 3, how it works. The inputs are slightly different. We have our proteins, which are still, or I'm sorry, they can be proteins which are going to be inputted as a string of amino acids. We have our DNA, RNA, ligands, or ions. Again, these ligands are small molecules, and the pair former, which is the brain, has it's simplified from the former on two, and it has reduced reliance on the MSA because it understands which atoms are near each other, and it works in atom based. But the really cool part is that the third step. It replaces the old structure module with a generative diffusion model. So diffusion, this is the same that we use for AI art. So we take random atoms, which is our noise, and then gradually we iteratively refine the atom coordinates till we get our 3D structure. And this turns out to be way faster and more accurate. And to do this, it's the constraints come from our pair former. Now, how does this thing work? Well, let's just briefly look over here. I'll bring up my AlphaFold server. Now, the server has let's just so we navigate to alphafoldserver.com, and you can either use your Google account or create a new account. Now it's going to take in, and we can create these jobs. We can, and here I've got this. I've already defined one of my inputs is a protein, and these are the amino acids that are in the protein. Um, and then I can also have as input RNA or ligands. And with the RNA, I would have to pass in a string of nucleotides here. The ligands, we can choose from things like GDP, ADP, And these are the small molecules. So there's a small list here, but then you run the job, and you can continue and preview the job. But I just to save some time, I had one or two here. So I'll just open the results, and this will show us the result. Those This is not a very exciting one, but oh, it's actually better. We've got the 3D shape. So this is a 3D shape of our protein and our DNA. And this is the predicted accuracy. So let me just switch back to my slides here. Um, And so we get results that will look like this. And here is a protein attaching to a DNA. Now, why would we want to do that? Well, this way the protein can modulate the DNA, like causing certain genes to start or stop. And the nice thing here in the gray of the stop at the gray is the what came out predicted from our crystallology, and the crimson is for our what came out of Alphafold 3. And the results are I think very good. So what can we? And this is just another. So with this tool, we can now because we can now show how a ligand, or this could be some kind of drug we're developing, a target drug. We can see how it can possibly bind to the protein. The protein's in the green, and the pink is our small molecule. And then we can keep iterating this, and keep trying to, instead of going into the wet lab and doing multiple experiments. So now this gives us the ability to explore undruggable targets, because many proteins, such as the ones causing Alzheimer's, have been labeled undruggable. Why? Because their structures were too complex or unstable to be mapped by X-ray crystallology. So AlphaFold 3 provides a high fidelity 3D map of these targets, revealing hidden pockets where new drugs could potentially fit. For example, here we have our disease protein, and AlphaFold 3 was able to show us where there is a binding pocket. And this is and then we can at the same time, it can model how a ligand can be docked into this binding pocket. And for some reason, these arrows are going the wrong way. But so where we can use this to design our ligand to attach to this protein. And now, just as an example here, we have molecular glues. Um, with molecular glues are interesting. We have these scopes in the cell. We have these scopes. The scopes are are going to float around, and that patrols the cell looking for old or damaged proteins. But in this case, the scope does not recognize this disease. And if it does see a disease protein, it's going to stick what's called a ubiquitin onto the disease protein. And this is really, and then along comes this kind of like a garbage collector, which is going to grab this disease protein and dispose of it, destroy it. But in this case, the scope doesn't recognize the disease protein. So we want to design a drug that is going to change this. Well, we're going to put this molecular glue onto the scope, and it'll give it a new binding surfaces that is able to bind onto this disease protein. So now, as soon as we have the chemical, we have this connection, there's a chemical reaction, and the ubiquitin is going to be released. Once this is done, of course, the disposal system can take over. Cool system. But what the hell is that got to do with AlphaFold? Well, everything. AlphaFold 3 becomes the architect because it can predict the three-way interaction between the ligase, the glue, and the target. And not only that, um, it can help design a neosurface on the scope protein, but it can map also aid us in mapping because AlphaFold is used to map all the 600 plus types of scope proteins in the human body. If we're going to do this by the traditional methods of the X-ray, it would be very expensive and very time consuming. Now we can move so much faster. Of course, we eventually have to do the verification in with the X-ray or the crystallology, but we've narrowed down things, and so the result we can reduce 4 years to approximately 18 months. So, example two, these CAR T cells. The CAR is chimeric antigen receptors. Is a form of immunotherapy, where a patient's own immune cells are re-engineered to hunt and kill cancer. So it turns a standard immune cell into a programmable search and destroy drone. Now, this was being done ex vivo for a few years, but it's very expensive. And it recently, in the last couple of years, has gone in vivo. It's going to be delivered through a lipid nanoparticle. But again, what has AlphaFold got to do with this? Um, back, whoops. Um, the biggest challenge in vivo is making sure the modification reaches only the T cells and not things like the liver or lungs. But AlphaFold, you can use it to design targeting ligands that are attached to the outside of the nanoparticle. Then AlphaFold 3 can model the interaction between the ligand and the CD3 receptors, thus recognizing the T cell. By simulating this handshake in AlphaFold 3, you can ensure the nanoparticle sticks to the cell long enough to dump its cargo inside. This delivers the genetic blueprints for the CAR receptor, which acts like the GPS, allowing the T cell to recognize specific cell. So, and then achieving the goal of being able to kill the cancer. So AlphaFold 3 has been just instrumental here in our progress with these CAR T cells, in particular the in vivo. But so very cool stuff. But in the context of modern biology, AlphaFold 3 is no longer just a standalone tool. It is increasingly being used as a functional module within an agentic AI framework. Now, what does this mean? AI with standard AI like basic chatbots, we're going to wait for the chatbot waits for a prompt and provides a response. Agentic AI is designed to pursue its goal autonomously. It can make decisions about how to complete a task without constant human hand holding. So we get way higher throughput, hundreds of interactions screened overnight. So here, all our design is on the computer. The agentic AI can use AlphaFold 3 to power a continuously self-correcting research cycle. As a hypothesis, an agent proposes a new antibody sequence. The agent sends the sequence to AF3 to see how well it binds to a target antigen. Now, then analysis, the agent evaluates the confidence scores, those the PL and DDT, which we discussed, and the binding affinity from the output. And then it keeps refining the results. If the binding is weak, the agent tweaks the sequence and restarts the process, doing in hours what used to take months of work. Oh, and this whole thing in here is going to, whenever we call, the agent is going to call into AlphaFold, it's using the AlphaFold API, an application programming interface. As a result, we have thousands, thinking it's almost like having thousands of human designers. working on a new molecule design. Just imagine what impact that would have on patients suffering from a rare type of cancer. The speed that we could get to a potential new molecule to address this medical need or ability to go after many diseases in parallel. Beautiful. So what does this do? The impact Alphafold 2 and 3. We take for target discovery, take our 1 to 2 years of crystallology. We reduce it to minutes for lead optimization. We had traditionally thousands of lab interactions, a lot of time in the wet lab. Here we have virtual co-folding simulations, and we drive our costs from millions of dollars way down. So AI-driven drug discovery has entered clinical trials with over 30 AI-designed or discovered candidates in human studies as of 2025, significantly accelerating timelines. And the key milestones like the phase 2 trials of in silico's uh, discovered a drug for pulmonary fibrosis and the first FDA-approved AI tool for clinical trials. So, wow, okay, threw a lot of stuff at you, but now we've gone from the gene sequence to amino acids to a 3D fold. This is the standard biology, and this is the way things have been done for 4.5 billion years by evolution. What if we could do the reverse? What if we could reverse this? What if we can go from specifying a desired function, work backward using algorithms, and generate a brand new sequence, and then synthesize a piece of DNA? Oh, what if we do this? Well, we can do this. Mother Nature, of course, has been doing this, as I said, for 4.5 billion years. And it's done a pretty good job. But I guess it can be argued how good the result is. But we've entered an era of protein creation. Uh, we have David Baker from the NIH Institute for Protein Design in the University of Washington. Now, have been the lead player here. Now, and in 24, that Nobel Prize in Chemistry went to Baker along with Demise and John Jumper. And what was his milestone? Well, they were cracked the code for proteins' amazing structures, shifting science from observing biology to creating it. And according to the words of the Nobel Committee, he succeeded with the almost impossible feat of building entirely new kinds of proteins. Ah, interesting. Maybe scary. So how do they do it? We have he has this RF diffusion program that's going to take a known protein and it's going to add some noise until it becomes a random cloud of atoms. Now, again, sounds familiar. That sounds like what Alphafold 3 was doing and what our generation does. So the AI is then asked to denoise this random cloud and using constraints such as a design a protein that has a flat surface to stick to a specific cancer marker. The AI then creates or dreams a 3D backbone that matches these requirements. And by the way, this backbone is not only it's accurate, but it's highly stable. But the backbone, the 3D skeleton, doesn't know which amino acids to use. And here, researchers use a tool called protein MPNN. And now it's going to figure out this is going to protein MPNN. It's a translation AI. It looks at the 3D skeleton and determines which of the 20 amino acids should go in each spot. So we come up with the amino acids to create this novel protein. Oh my. So now we've got the amino acids. Well, if we have the amino acids, we know the nucleotides required for the DNA to produce the gene that will produce this protein. So M coming into the DNA synthesizer is the machine we're going to feed this input into. And think of this as a benchtop printer. It's going to receive the digital genetic code and it's going to print the custom DNA. And now we do have machines like the enzymatic DNA synthesis. EDS. It's like building with Legos. The printer just adds one brick, a nucleate, or think nucleotide, washes away the excess, and unlocks the next spot and repeats. But what comes out of here has to be in a soluble form because of what that we will see in the next step. So we print the DNA, but it's going to be print in a soluble form, and then um, then we're going to take and we're passing it into this cell-free solution. For a few years ago, they were pumping this into E. coli bacteria itself, but the E. coli sometimes rejected our synthetic DNA. But what they do now, they build up this cell juice and cell free. There's no cell. They break up the E. coli membranes, and we're only going to leave over the protein producing parts, the ribosomes and the polymerases, and they're going to harvest this and now, because there's no cell here, this immediately starts working, and starts the moment but we have to first add amino acids. Why add ATP? ATP is for the energy, and of course, because we have our mRNA, the DNA and the polymers are going to produce our messenger RNA, and it's going to be copied into and it's going to want to have the corresponding amino acid. So we have to pour in and make those amino acids available. But as long as they're in our solution, then we're able to, and because this thing is cell-free, the process starts almost immediately. So this process happens fast, and we get in 2 to 4 years, bingo, bango, we have a novel protein. And then the final protein is extracted and validated, often faster than living cells do. So what's this good for? Well, we have the first de novo medicines during the pandemic. The Institute of Protein Design developed a fully synthetic COVID-19 vaccine, the first de novo design medicine approved for human use. So as a case study, we have a generative immunology. And the way this works is for decades, we're going to bypass the animal immunization to engineer binders from scratch. And for decades, antibody discovery was more of a guess and check process involving animal immunization or massive library screenings. But then we switched to de novo design, where antibodies are built atom by atom on a computer to hit specific predefined targets. And if this is our antigen, this can be anything from a cancer, and we have hotspots, or with the hotspots, identified hotspots, which we want to bind to with our drug. And so the way we do this is we have our target selection. The backbone of these are always they're constant, so that on the ends of these are CDR loops, and these are the flexible parts that can be hit different cancers. And so what we did, we identify the hot spots, and then we do our backbone generation using that RF diffusion program. And it's going to the main body of the antibody is constant, and we so don't do anything with it, but we denoise these CDR loops because these are the variable parts, and this is where we want to create a novel CDR. So then we do our sequence design using protein MP and N, and of course the framework part remains fixed, and then we generate our flexible, our variable CDR loop, and then we have our new antigen. So, and then we can print it on DNA or whatever we want. So then we have a novel antigen. Um, Kind of like we did back in with Alphafold 3. But what is other applications here? There's not just medical applications. We can take and we can break toxic bonds. Nature never evolved to break down synthetic pollutants. And AIs generate enzymes designed specifically to degrade forever chemicals like plastics. So we're helping Mother Nature out here. And then for in the atmosphere, designing entirely new biological pathways to fix carbon and actively remove methane from the atmosphere. Of course, not everybody believes this needs to be done, but at least we have the option. And then again, for the rising temperatures threaten global food security, at least according to some people. And de novo design is increasing the stability of proteins in major crops like rice, enable them to thrive in hotter and hotter climates. So the future of biology is intentional engineering. So we can go from having our from the computer been totally in silico, and we can dream up new proteins. We have a new superpower. We are leaving behind the area of random mutation and screening. This is evolution. And with deep learning and computational design, biology is becoming a programmable engineering discipline. Super interesting. Now, but of course, Mother Nature took 4.5 billion years with evolution, and now we have something that we can do comparable in 4 to 5 days. Well, are there safeguards? Yes, there are safeguards. There are some safeguards. We have scanners now break the requested DNA into tiny fragments as small as 16 amino acids and check if those fragments are parts of known toxic proteins. Well, yeah, but if we're creating a de novo DNA, this is not going to be in that database, and it'll do some red flag list. If a sequence is on the edge of being suspicious, the printer will hold the job until a user provides a legitimate research justification. Yeah, well, that's well and be. But the bad guys, they're not going to follow these rules. They're going to print whatever they want. Anyways, um, I hope this provided you some information to what I think is such an exciting field, combining everything, the two big things, my AI and my and medical interest. And again, it's this is just a start. We're evolving. All this stuff is moving so fast. And but if you're interested, like anything else, you should in our older age with more time, we should never be bored. There's so much we can learn, but diving in to using like Gemini and Notebook, LM, you keep asking questions and you keep getting more knowledge and more information. It's wonderful. Stay curious.